Good morning, everyone. Thank you for being here. It is my deepest honor to introduce you to Dr. Will Eward. Dr. Eward is an associate professor of orthopedic surgery at Duke University, and he is an orthopedic oncologist as well as a veterinarian. And he's a strong champion for comparative oncology research, and he was one of the leading forces in creating the consortium for canine comparative oncology back in 2016. It was a consortium that started between Duke University and NC State, and it's blossomed since. And Dr. Eward's research, seeks to answer many questions surrounding sarcoma for both children and dogs, and other species as well. And so without further ado, I'd like to introduce you to him and his talk this morning, which actually has a wide range of topics. So I won't try to summarize it, but I'm sure it'll be very enjoyable. So join me in welcoming him, please. Thank you.
Thank you very much. If this title slide looks chaotic, it's because this talk is chaotic, and that's by design. I hope you won't hate me for it. You will not hear me say words like background, introduction, methods and materials, results. What I really want to do is just tell you a story. And personally, if I go to a very specific conference and I'm in a session on something very specific, I want to hear a very specific scientific talk. But when someone visits us to present a seminar, I hope that they will, above all else, make it entertaining and interesting. So that is what I hope to do. And I still hope to give you some things to think about and to tell you a story about how we think about cancer across species, cancer in animals, and how we integrate that into our broader goal of making things better. So first, thank you very much for this invitation. I had a lovely time at dinner last night and meeting people here, and it really is an honor to be here. And especially thank you to Dr. Joanne Tui for making this happen. If anyone doesn't know her, you should. I've learned a lot from her over the years about immunology and histotripsy, but I've also learned a lot from her about being kind and patient and compassionate. So you just have someone really special in your midst. And someone was asking me last night how long I've known Joanne, and I thought, well, I at least knew her while she was getting her PhD, and then I thought, well, I also knew her when she was a resident, and you can see from this picture she looked exactly the same. And I thought, I think I actually met her when she was a student, before she even had a DVM, and so I have a picture of her from that era. And she really hasn't changed. And then I was looking for a picture of myself from back when I met Joanne, and I realized I've actually changed a lot. So, you know, the career can take a lot out of wear sunscreen. Now, I am very, very thankful to be an orthopedic oncologist, but I'm also really, really thankful to be a veterinarian. And I found it kind of traumatic crossing from the veterinary world to the human world, just because the culture is so different. And I came into the human world really, really keen to studying sarcomas at a higher level. And I had people ask me a lot of crazy questions like, do dogs get cancer? Or do you have to scrub your hands to operate on a dog? So it was something traumatic. And when people ask me why I switched, I never thought of it as actually making a switch. I had a patient who really impacted me, a golden retriever with an osteosarcoma, that led me to want to do more. And for those of you looking at this radiograph on the right, many people will look at this and say, oh, here's this bone-forming lesion with an ugly periosteal reaction. And many people will look at this and say, that's an osteosarcoma, before they even recognize that it's not a person, that it's a dog. And there's a good reason for that we'll talk about. So I do like kind of being in both of these worlds. And my heart does really straddle these two worlds because there are things I like from the human medicine world and things I like from the veterinary medicine world. And I really like the operating room, and the human operating room, it just has seemingly unlimited resources. And the veterinary operating room usually has more interesting things happening in it, and you have to creatively figure out how to do them on a tighter budget. The main difference is that the patients are much cuter in the veterinary world. So this is my wife, Cindy. Cindy's the best surgeon I know, other than Joanne. And for any MDs or basic scientists, when we think about human surgeons, they're very, very specialized, but veterinary surgeons do it all. And so I like being married to a fellow veterinarian, but when you have two veterinarians married and living together, you just accumulate animals. People just bring you animals. So you can be having a nice vacation in Florida with your family, and then next thing you know, you hear your mother saying, an injured sea turtle, my son and his wife are veterinarians. And then you're in a crappy kayak trying to capture a sea turtle in a plastic bag, put that sea turtle into your little rental car while kids come out of the woodwork to go into the trunk and pet it. So Cindy and I decided to just embrace this. And we set up a farm that if you were driving by, you might say, oh, that's a proper farm with sheep and horses and chickens. But actually, the truth is, none of the sheep are for eating. The horses are only kind of for riding. And it's really a bunch of reject animals that we've given a home to. I actually have a sheep with lipomatosis, which people can say. And at any given week, we're probably bottle feeding something or dressing wounds or just caring for something that's been brought to the house. This is probably about a third of the current population of animals living in our home. So anyway, on to the story. Chapter 1 is the big picture. And you might say, well, why should we care to hear a talk that has a lot in it about dogs and other animals? And the reason for me is that we can learn different things. We can learn things in different ways. Or in the case of sarcoma, we can learn things with a different incidence. So sarcomas are very, very rare in humans. If you look at osteosarcoma, there are about 1,000 human cases a year. In dogs, we don't really know what that number is. I think it's probably close to 75,000 a year if you were really capturing them all. Now, I've got 2 human patients in this talk who've both given me permission to show them. And so you won't see anything covering their eyes, and that's by permission. And the first one is this girl, Kay. And Kay presented as a 12-year-old with knee pain after basketball practice. And you can see here what's a very classic appearance for osteosarcoma of the proximal tibia. And this is Sheba. Sheba was a 12-year-old lab at the time I met her, and her lesion is a little harder to see, but she's got a similar lesion in her distal radius at the top of this screen with a pathological fracture running through it. And for both of these patients, the treatment is some combination of surgery and chemotherapy, hopefully. Now for Kay, she's a person, so there are just vastly more resources for this. One luxury we have is we're able to get an MRI of her tumor, not just once, but we actually get one before chemo and then after chemo to really plan out the surgery. And if this second picture, it looks like I've photoshopped out a little black box, that's actually where during chemo, the tumor just started fungating through her skin. And there also are seemingly unlimited resources during Kay's surgery. So this is a standard proximal tibial replacement in a person. And this implant you're seeing here, this is the trial version of that implant that we use to get the size right to reconstruct her skeleton. But this implant, the cost to the hospital is about $28,000. And you don't even bat an eye at that in a person, because the results are very good. This is what Kay's leg looks like at the end and what her radiographs look like. And she'll go on to get more chemotherapy. And then this is her walking you can see in the video, this is even before her hair has grown back in from chemo. So you get really, really good outcomes for the money you spend. Interesting side note on this girl, she went back to playing basketball with this prosthesis, and she actually dunked a basketball off a trampoline, broke the prosthesis, had to have it revised, and went back to playing basketball again. Now, things are a little different for Sheba because the resources are very different. And most dogs, as many of you know, can have an amputation and do quite well. We often say they're three-legged with a spare. Sheba, for a variety of reasons, she had some old dog vestibular disease and cruciate disease, and she would not have done well missing a limb. So I'll just show you one of the simple veterinary versions for limb salvage. This is a plate that costs about $750, made by a company called Veterinary Orthopaedic Implants. And this is a block of stainless steel that has holes machined into it so that you have a bridge plate that's screwed into the bone above and below, and then screws that go into this block of steel. So it's a very simple, very cost-effective implant. And this is what Sheba looks like when she's healed. And this is Sheba walking around. So it's nowhere near a normal gait, but it works. And my point of showing these to patients is to suggest that these are two beings with one cancer, and that osteosarcoma is a great example of something, of a disease state that is really two sides of the same ugly coin when you look at it in dogs and you look at it in humans. In both cases, you have a painful mass. In both cases, you have a bone-forming lesion that oozes through the Haversian canals. You have an ugly periosteal reaction. And if you're a pathologist with microscope slides of canine and human osteosarcoma, you certainly cannot tell which is which. And if you have a lab, meaning a laboratory, not a Labrador, you also cannot tell. So this is a classic paper from 2009 looking at RNA sequencing data from human in yellow and canine in black osteosarcoma patients. And what these authors showed is that all of the ways you can cluster these sarcomas together, so all of the high-grade ones cluster together, all of the chondroblastic ones cluster together, the one way they don't cluster together is by species of origin. Now, these are some survival curves for patients presenting with localized osteosarcoma. And in the old days, before we knew the value of chemotherapy and before we had chemotherapy, almost everyone died. And then in the 60s, people started using cyclophosphamide and things got a little better. By the 80s, we were using combination chemo and things were a lot better. But if you look at this curve from 2004, it's really about the same as the 1980s. And if we did one in 2024, it would be the same. And that's depressing. Also depressing is when you put the canine curve on here. If you're a dog and you're diagnosed with osteosarcoma, you are almost certainly going to die from osteosarcoma rather than something else. But on the other hand, if you have more canine patients with this disease and they are likely to die from it, usually in about a year, even with treatment, that's probably the species in which you should be studying it. So for the minority of people in the audience who remember the 1980s or were even alive, you might ask yourself, what was the same in the 1980s? And the answer is not very much. The only thing on this slide that's really still relevant today is this picture of Mr. T. For you kids, that's Mr. T. You should Google him. So I just want to mention a couple of terms that you'll hear throughout the talk that probably most people here are familiar with. But one medicine is the idea that health and disease are health and disease, and they're not species specific. And comparative oncology is the idea, it's a subset of that really, that we can compare same cancer types across species. And this is something that has become popular in the last 1 to 2 decades, but it's not a new idea. And almost everyone knows Rudolf Virchow, Virchow's triad, Virchow's node, the man who dragged medicine out of the Dark Ages in the mid-19th century. And there are a lot of Virchow quotables. If you Google Rudolf Virchow quotes, you'll see a million really wise things he said about medicine. But one thing he said that was largely ignored was this: between animal and human medicine, there's no dividing line, nor should there be. No one seized upon this in the 19th century. They sort of said, I don't know about that idea, Rudolf. Okay, so I promised there would be a little bit of chaos, and I don't know what everyone in this room or on the Zoom is interested in, so I'm going to punctuate this talk with non-cancer things and some hopefully relevant animal stories. The first one I'll tell is about a dog named Shaq. This is Shaq. When I met Shaq, Shaq was a 12-year-old golden retriever. And I actually get chills when I tell this story. I probably wouldn't believe this story if I were hearing it, if I didn't know it to be true. It's a true story. And Shaq presented with a rib osteosarcoma. And kind of an unusual owner and unusual request. Shaq was given to a 12-year-old girl by her parents as a present when he was a puppy. And that 12-year-old girl is now 24 years old and coming back to her childhood home for her wedding. And so Mr. Sundram, Shaq's owner, comes in and says, I want Shaq to be alive for the wedding in 6 months. And I want my daughter to not know that he's sick. So we said, well, tall order, but we'll see what we can do. So we did this chest wall resection. This is the defect in the chest wall filled with just simple, cost-effective polypropylene mesh. We swung a latissimus flap over it, closed the skin over that, over some closed suction drains. And this was Shaq just a few weeks later. And everything seemed to be going well. And about 6 months down the road, I got a message that Mr. Sundram wanted to meet in person. And so I met him and I went into the exam room and he was there and Shaq was not, which seemed bad. And he said, I wanted to thank you in person. Shaq was alive and well for the wedding. And as my daughter and her new husband drove away in the taxi to start their honeymoon, my wife and I were waving goodbye and Shaq was there with us, wagging his tail. And the taxi turned out of the neighborhood and Shaq dropped dead. So, you know, it's often said we don't deserve dogs, and there's just so much about them that we don't understand. But I've been around dogs long enough to know it's entirely possible this dog would view his job as being to stay alive. So the bigger picture, have you ever asked where cancer exists in the world or why we even have cancer, why we haven't just evolved out of it? And if you're looking for a very long but interesting paper that will at times wake you up and at times put you to sleep, I would recommend this one. Athena Actopus is a brilliant biologist at Arizona State University, and she spent years crafting this paper called Cancer Across the Tree of Life: Cooperation and Cheating in Multicellularity. And she makes some really thought-provoking points in this paper that I'm just going to put up on the screen. So she taught us in this paper that multicellularity as a concept didn't just evolve once on planet Earth, as I would have thought. It actually evolved through 7 separate channels. And across these 7 separate evolutionary channels, there were some common things. To be multicellularity, you had to evolve the ability to suppress cell-level fitness to promote organism-level fitness. And she describes cancer as a breakdown in this multicellular cooperation, with the cancer cells cheating in ways that can be devastating to the organism. And she says to be multicellularity effectively, you have to suppress that somatic cheating and the cancer that results from that cheating. So has anyone heard of Peto's paradox? You don't have to actually raise your hands. I always hate that. It's just a rhetorical question. But there was a biologist English biologist named Sir Richard Peto, who made the very correct argument that cancer should be somewhat mathematical, that any given cell should have some mathematical probability of going from healthy to cancerous. And so if you think about these three beings, the human has at least 100 times more cells than this mouse, and this elephant has at least 100 times more cells than this human. So mathematically, you would expect the elephant to get the most cancer and the mouse to get the least. But in fact, it is inverse. You can, and maybe you will, make a lot of comments about mice and cancer, but you can put whatever three organisms in here you want. In general, the bigger organism will get the least cancer. And that is Peto's paradox. But I'm not sure we should view it as a paradox. This elephant was on the cover of Newsweek in 2015 or 2016 because he really illustrates what evolution does with multicellularity. It says if you're going to have a large body and a lot of cells, you have to evolve ways to stop that cheating. And the elephant became famous when a group from the University of Utah published their findings on elephants having 13 copies of P53. So how do we use our knowledge from other species to learn about cancer? I'll tell you in a second because we're going to have another story. So this is Kira the deer. And there are lots of cute animals out there, but none have stolen my heart like Kira. Public service announcement, deer have evolved so that fawns have almost no scent. And so the mothers intentionally leave them so that their scent doesn't attract predators. They leave them alone and they come back periodically to feed them. So there's actually a problem with people finding a, quote, abandoned fawn and bringing it in for rescue. It usually does not need your rescue. Here it was a little different. I was on call operating on a Sunday and I came home and my teenage son says, Dad, some adults brought this injured deer to the house for you and Mom. It's in the barn. And I found this very new fawn with a moist umbilical cord covered in dog bite wounds. And as soon as my daughters got home, they relocated Kira from her very nicely made home in the barn to their bedroom. And we said she probably will not survive this, but we can try. And she did, in fact, survive, drinking bottles of goat milk Q2 hours and living upstairs in the bedroom. So my wife's parents do not like animals, which is funny because two of their kids are vets. They think we're crazy to have all these animals in the home, and they always lecture us about this. They've never had pets, not even a goldfish. And so my wife calls the family together and says, Grandma and Papa are visiting this weekend, and I do not want to hear about having this deer in the home. So for their 3-day visit, Kira stays upstairs. Nobody talks about her. And we all take turns going upstairs to bottle feed her. And everyone said, okay, Mom, great, we got it. So the grandparents arrive and we're having dinner and everything seems good. And I look over at my father-in-law and I think he's choking on his food. And he starts pointing and he says, there's a deer in your house. And he said it the way you might say, there's an axe murderer in your house. And anyway, there indeed was Kira. She had escaped the bedroom and was walking down the stairs to find out what was happening. And anyway, this man who doesn't like animals was instantly charmed. And for the next 3 days, he wanted to be the only one to do all of the bottle feeds. So how do we use that knowledge from other species? I'm going to give you just a broad overview with a couple very shallow dips into some of the rabbit holes about how we do this. So one of the things I really like about my job is we have this comparative oncology group. And one of the best tools in our toolbox is what we call the MDH, or mouse, dog, human pipeline. And that essentially starts by collecting sarcomas from lots of different species. I need to update this slide because this is not all of them. Now, I do think if you have a sawfish on your slide, that's sort of a PICS or it didn't happen kind of thing. So this is a sawfish from Ripley's Aquarium in South Carolina that had a sarcoma. Here it is after removal. I'm happy to talk after the talk about how you anesthetize a sawfish and also some of the quirks of growing sawfish tissue in cell culture. And then, you get a pathology report no matter what your sarcoma comes from. And I was really sad to see that this sawfish doesn't have a name. So Ripley's needs to name their sawfish. So anyway, we start with patients. And the first thing we do is grow PDXs, patient-derived xenografts, because that's easy to do. And then we try to establish cell lines, organoids, and microorganispheres. This is Lori Graves. She's our newest hire. She's a pediatric hematologist oncologist. And she's really done all of this work with the bulk organoids and the microorganispheres. These are bulk organoids on the left, and these are microorganispheres on the right. And these are, it's sort of a partially automated process where you extrude cells into a lipid droplet. And it just gives you a kind of easier to work with, more uniform quantum. And the next thing we do is we do high throughput drug screening, usually on the cell lines, also on the organoids. And we've learned some really interesting things on the organoids. So first of all, the patient-derived osteosarcoma organoids, and this is a bunch of osteosarcoma organoids from different dogs, they model inter-tumor heterogeneity in response to chemotherapy. So these all get treated with doxorubicin and a platinum drug, which are the drugs we use in real life. And we see this heterogeneous response in how these respond. And it's interesting to me, if you look at these yellow ones, and to a lesser extent, the green ones next door, how sometimes we'll have two different tumors from two different individuals that have almost an identical response. You would really expect this to be even more heterogeneous, or I would. And then the other thing that Lori does is she collects and grows organoids from different parts of the different tumors. And so these organoids also model intratumoral heterogeneity, where you see differential responses from organoids started from different parts of the tumor. And again, we see the same thing where sometimes in this one example, piece 5 and piece 9, these came from very remote areas of the tumor, and yet they seem to be very similar clones, at least in terms of how they respond to therapy. And then if you look at RNA sequencing data from the doxorubicin-resistant organoids, which are probably the parts of the tumor we care about, the ones that are really chemo-resistant, these are probably the clonal populations that are going to metastasize and kill the patient. And we actually do tend to see upregulated gene expression in a number of targeted pathways. And so we also will do whole exome sequencing, and then we ideally would take this information back to those patients, or at least refine our precision medicine approach. And the overall workflow looks something like this. So just to give you one example of the kind of output we get from this pipeline, this is Alex Lazarides. Alex was with us as an undergrad, as a medical student, as a resident. He's now an orthopedic oncologist at Moffitt Cancer Center, and he did so much good work when he was with us. But he wrote a paper called A Cross-Species Personalized Medicine Pipeline Identifies CRM1 Export Pathway as a Potentially Novel Treatment for Osteosarcoma. So the first thing that Alex did was he did this cluster analysis of in vitro drug responses just using some standard human and canine cell lines, and showed that these responses are fairly uniform regardless of species. The next thing he did is he started with a 119 oncology drug compound screen using human and canine osteosarcoma cell lines. So on these plots, dogs are plotted on the x-axis and people are plotted on the y-axis. And you see for both this 119 compound screen and this 2100 compound screen that you have quite a good correlation. And then this drug screening identified a number of pathways as potential targets for osteosarcoma. And you may say, well, what about those other pathways? This is just highlighting some of them. Some of them also turned into other areas of investigation, like the proteasome. Other of these, like kinesin, aren't really that easy to drug. So they're just sort of sitting on the back burner. But certainly for CRM1 targeting, he was able to show a response to inhibition of this. So what is CRM1? CRM1, also known as exportin, is a nuclear transport or nuclear export protein. It's a protein that helps shepherd things out of the nucleus and into the cytoplasm. And it is ubiquitous, but in many cancers and in almost all sarcomas, overexpression is very, very common. And in fact, if you look at osteosarcoma clinical patients, high expression of CRM1 is a poor prognostic indicator. So in some in vivo studies with tumor-bearing mice, Alex was able to show that indeed, as we would have predicted, treatment with a CRM1 inhibitor was actually effective. And so this is the type of thing that we hope can then generate interest in our colleagues on the human hematology oncology side to trying this in human osteosarcoma patients. Okay. In case you're getting scientifically overwhelmed, next animal story. I will tell you, my wife and I are big dog people. This is the first small dog we ever had. This is the first dog we ever had with a personalized name tag, and the first dog we ever had who wore a sweater. And when I told my kids I was preparing this talk, I said, I need some cute pictures of Pete. And they sent me this, which that is a pretty cute picture. They also sent me this. And I don't know what the story is here. But Pete is alive and well. And so Pete's original owner was an elderly lady with dementia, and she put these like bracelets on his limbs, and he got necrosis of his toes. And he's a good boy, so he ate the necrotic tissue. And what you're looking at here are close-ups of Pete's feet. So we moved his carpal pads distally to give him something that he could walk on. And I wish I'd gotten pictures of that surgery. The closest thing I had is this other cute picture of Pete when he still had some granulation tissue around where we pulled those down. And Pete is still barely a kilogram, and he was even smaller as a puppy. And it took him many, many months before he could actually use his forelimbs. And so we forgot about disuse osteopenia, your bones getting weaker. And as soon as he started walking, this happened. This happened on Christmas Eve. And we actually don't keep splinting supplies at our house, but he needed a temporary splint. So if you have kids and it's Christmas Eve, what do you also have? You have Legos. So this is a splint made out of Legos. But then he still needs some definitive fixation, and he was too small for even like the mini frag plates. So this is an external fixator. And if you're used to looking at external fixators, except for veterinary surgeons who know well about this trick, you may think this looks funny. And what this is it's these little wires called K-wires and anesthesia tubing. And the anesthesia tubing is accordion shaped. So you put the K-wires through the tubing into the bone, you fill the tubing with polymethyl methacrylate, and then as it polymerizes, you can hold the limb out to length. It makes a perfectly good external fixator. And then here is Pete, much later, chasing Kira. So the next chapter is Pete's theme, only bigger. And this is in case there are surgeons or biomedical engineers in the audience. We'll take a break from the lab aspects of cancer. So this is a human patient who presented as a 57-year-old man to the emergency room in fulminant heart failure with an ejection fraction of 13%. That means your heart is not pumping out a lot of the blood that comes back to it. And he just casually says, I haven't been feeling well all week. And this is the only consult we've ever gotten for this indication. The cardiologist said, while performing a catheterization, we have glimpsed something in the femur. Here's what they glimpsed. Here's a bigger view of this. And here's an MRI. So if you're thinking there's this permeative bone lesion that's sort of growing in a multilobular fashion and is relatively bright on a T2 weighted MRI, most people wouldn't know this, but if you're a sarcoma nerd, those are all the features you would expect of chondrosarcoma. And chondrosarcoma is notoriously refractory to adjuvant treatments like chemo and radiation. It's typically best cured with surgery alone. But I would not want to replace the entire femur of someone whose heart barely works. So we decided to try treating him definitively with what is a very high dose of radiation to his entire femur. And so everyone could live with this, and we thought this man probably doesn't have long to live, and this should take care of things. But then there was a plot twist. He comes back into clinic 3 months later out of the blue, and he looks great, and he feels great, and he says, I can walk for miles, I can walk upstairs, and I stopped taking my heart meds. So if you're asking yourself what causes severe cardiomyopathy that can reverse spontaneously, you may also ask, hey, did this happen during the pandemic? And it did. This was actually an example of someone with viral cardiomyopathy that can just correct itself. So now we have a real dilemma because we've treated him with a treatment that we wouldn't expect to work. And now instead of looking like someone who's going to die in the short term, he looks like a pretty healthy guy. So we had this big multidisciplinary discussion that somehow resulted in me getting nominated to replace his entire femur, which I was very nervous about doing given the prior radiation. And side note, here is his tumor after it was removed, and he only had about a 70% response to the radiation. So the radiation would not have saved this man's life. So it made us feel good that we did this. And I was so worried he would get infected, but everything just seemed great. And then he comes back for his 3-month follow-up like this. And you can almost sort of see it, but you can tell this femur has been dislocated for a long time because he's starting to get a neoacetabulum way up there at the top of his ilium. So the ball is nowhere near the socket, and you're never going to be able to pull it back in when it's been out for a while. So truly a great sadness. And initially I thought, well, maybe he could just live like this. It's still better than being dead, but he could not. And every time I saw him, he said, please, you have to fix this. I cannot live like this. So you may ask yourself what you would do now. We thought, well, maybe we could design something creative that would allow us to operate above the radiation field and put the ball into a new socket that's way up above where he's been radiated. So we designed this little plastic capture for a reamer where we could make a hole in his ilium way up high. And we designed this prosthesis that we just called the pocket prosthesis. And we 3D printed it. Here it is coming off the build plate. And then here we've pulled this little flap down way up around his belt line. This is the capture guide in place for the reamer. There's a reamer going through it to create a little hole. This is that pocket prosthesis up in his ilium. And then now we've connected it to the femur so that we didn't need to pull the ball down to where the socket was. And this worked really, really well. So good we did it twice. Yes, we did. So we also had this 21-year-old man who had a hemipelvectomy that removed his hip joint and the lower part of his pelvis. And he had initially elected to be left what we call flail, meaning no reconstruction. And 3 years down the road, he was really, really unhappy with this. He had been a gymnast and a dancer, and he really was unhappy. He said, can we go back and reconstruct my pelvis now? And I would have said no, but after our experience with this pocket prosthesis, I said, sure, have we got the thing for you? So here is that little capture guide again. Here is his little pocket prosthesis up in this upper part of his pelvis. Here it is from the front. And we put a fairly standard hip replacement stem into his femur, and we connected it. And here's where it gets crazy. This person comes to see me from very far away, so I don't get as much follow-up with him as I would like. And the follow-up I do get usually comes in the form of TikToks. So on Halloween, literally on Halloween night, he sends me this TikTok of him as Medea at a party. And I have to fast forward before this video ends because there's something obscene at the end that I couldn't edit out. And so I said, Kayvon, you seem like you're doing pretty well. And he said, well, I'm better than I was before, but I still really struggle with dancing. I said, okay, well, can you send me a video and show me what you're having trouble with? And he said, it's the heel toe. I'm going to just turn the volume up. So anyway, in my view, Kayvon is doing quite well. He certainly dances better than I ever have. So I think we'll continue using this prosthesis. So how do we filter this cross-species information? Well, sarcomas don't have a high mutational burden compared to things like melanoma and lung cancer, but they still have too many mutations in genes to really effectively study. So how do we take all this data we get from different species and try to identify relevant genes and new targets? Well, one of the workhorse things you need are synteny maps so you can compare across species. And this is an example of a dog to mouse synteny map. So the dog's 38 autosomes are shown here with these solid colors on the right. And then when you map where that genetic material is found in the mouse, you see this mosaic appearance of the mouse's 19 plus XY chromosomes. And so we make a lot of synteny maps for our different species. And then I'll just show you an example of a three-way comparative analysis of canine, human, and murine osteosarcoma. So we start with 14,000 genes in common, which is obviously an unwieldy number, to 117 specific copy number variations that are common to canine and human osteosarcoma, which you could still spend a career ferreting out 117 copy number variations, to then when you add in the mouse, only 5 shared copy number variations. And if you say, well, what are those 5? The first 4 of them are not very interesting. If I said to my boss, I did all this work and guess what, P53 is important, I probably wouldn't have a job. But one thing that came up was the ATRX gene, which really was at the time not on the radar as being relevant in sarcoma. ATRX is responsible for this condition called alpha thalassemia retardation X-linked syndrome. And it is relevant in gliomas, but it really wasn't on the radar for sarcoma. So this is Suzanne Bartoff DeWitt, and she actually did her PhD exploring the role of ATRX loss in osteosarcoma. And the first thing she did is created a nice genetically engineered mouse model where she showed that floxing out ATRX would result in an increased rate of tumor formation. She also showed that knockdown or knockout of ATRX would result in faster growth of these tumors in a flank model, a patient-derived xenograft model. She showed in scratch wound assays that there's an increased rate of wound closure with knockdown or knockout of ATRX. This all gets done in a variety of different osteosarcoma cell lines. And then she showed in some different migration and invasion assays, when you knock out or knock down ATRX, you also see an increased ability for these malignant cells to migrate and invade. And she then went on to show that these cells that have lost ATRX have a therapeutic vulnerability to integrin inhibition. And you might ask yourself why. A lot of this is hypothesis generating rather than known, but there actually is a kind of intuitive reason why this would make sense. And so she showed that if you treat these mice or these cells with an integrin inhibitor, it's uniquely effective against this subset of osteosarcomas. And actually, if you provide integrin inhibition and then you repeat those scratch wound studies, you sort of normalize the growth of these cells across a scratch wound. So she went on to show that ATRX causes an upregulation in NF-kappa B signaling pathways. And what's interesting about that is integrins also cause an upregulation across NF-kappa B signaling pathways. And that has the downstream effect of making things more aggressive, and integrin inhibition reverses that. So I'm nearing the end. the last real chapter is just kind of some anecdotes about large, dangerous animals. So this is Alice, the white rhino. Joanne, did you know Alice, the white rhino? This is a rhino at the North Carolina Zoo who needed a biopsy. And she's an incredibly sweet animal, but she also weighs 5,200 pounds and has poor eyesight and a horn. So even just her trying to come up and say hi to you can be very deadly. So how do you anesthetize a rhinoceros? And this is not a dad joke where I say very carefully, funny as that would be, but actually you need some really potent drugs. So an animal like rhinoceros will be darted down using carfentanil and atorphine. And if you're not familiar with carfentanil, carfentanil is like a cousin of fentanyl, but it's 10,000 times more potent than fentanyl. And so before we even open these drugs and load the syringe, before we even open the outer box that these drugs live in, everybody has to put on PPE, because one drop of carfentanil on your mucous membranes and there's not enough Narcan in the state to save you, you will just die. And then I've also had a lot of fun operating on some bears over the years. I may save the story for Willie. There are actually a lot of stories you could tell about Willie. But I was contacted by this vet from a zoo in Alabama, probably a slightly sketchy zoo. And it was a vet that I knew from vet school, about Buddha the black bear who had swallowed a balloon. And so I roped my wife into this and we drove down to help remove this balloon from Buddha's stomach. And we didn't know much about Buddha, but Buddha got the name Buddha because he likes to eat and is fat and he looks very cute and fluffy. And we thought maybe Buddha's nice. And so Buddha gets anesthetized. We do a successful laparotomy, a gastrotomy, remove the balloon, close Buddha up. And now it's time for Buddha to go back to the bear enclosure. So Buddha gets loaded into the back of this van. We pile in with a bunch of the zookeepers and Heath, the zoo vet, and some guy named John who's riding shotgun. And Heath says, all right, everyone, I want to go over the plan. I'm going to drive really fast toward the bear enclosure. And if Buddha wakes up, Jenny, you open the back door to the van, everyone jumps out, and John, you're going to shoot Buddha. And I thought, that doesn't really seem like it qualifies as a plan. It seems like what you would do if you didn't have a plan. Anyway, fortunately, Buddha was sound asleep and we got him back to the zoo enclosure. So last thing I want to say on this topic of large animals is kind of a funny story. This is B. Schloopner, Nicole Levine, and Buddy the elk. And if you look closely at Buddy the elk, Buddy's the one on the right, you can see Buddy is growing some new bone out of an area where his antler velvet has been amputated. And 2 years ago, there was this unpopular decision made at Duke that we were going to have grand rounds on December 23rd, 2 days before Christmas, and that some residents would be assigned to give those talks. And nobody was happy about that. I wasn't happy about that. Nicole Levine was one of the residents assigned to give a talk 2 days before Christmas. So as a form of protest and humor, she wore these outrageous reindeer pajamas and found a way to incorporate reindeer into her grand rounds talk. And after the talk, she says, Will, I was reading about reindeer and I'm really mystified by how these antlered animals just grow new antlers every year, but don't ever get cancer. If you've never stopped to think about that, so antlered animals, the photoperiod triggers them to grow their bony headgear. And it starts out as this soft tissue called velvet, which is just ripe with epithelial to mesenchymal transitions. And it does all the things that cancer does. It rapidly divides, it invades through membranes, it calls in new blood vessels to serve it. It's actually the fastest growing tissue in the animal kingdom, but you never see cancer of antler velvet or antlers themselves. And then in the spring, when the photoperiod increases, these antlers get shed and the whole process starts all over again the next year. So Nicole single-handedly got us thinking about this purely by her reindeer-themed protest of grand rounds. And she contacted an elk farmer in Western North Carolina. And we went out and we resected a little bit of the velvet from Buddy the elk's growing antler. And then we started growing some cells from it. And there's actually enough published on this that you can use immunostaining to make sure that you actually have the stem-like parts of the antler velvet. And then we did quantitative proteomics to compare a few things. We compared the bony parts of this, what we call EAC, elk antler cells, to the soft tissue velvety parts, EVT, antler velvet, and then 143B human osteosarcoma cells, a mouse osteosarcoma cell line, and then normal mouse bone. And if you look at this unsupervised cluster analysis, what you see is that the elk velvet tissue is at one end, next comes the bony cells, then the two cancer cell lines, and then in a completely different clade, the normal bone. So that's really fascinating to me that this growing bone would not classify itself in the same clade as the actual bone. And what B went on to show is that the elk antler cells are actually enriched in P75 receptors, MET signaling, and they're also enriched in P53 and P53 adjacent pathways. So I think P75 makes sense because that's an osteogenic differentiation pathway. MET is interesting. We associate that with chemoresistance, metastasis, migration, invasion, things you would need if you're going to grow bony headgear right out of your skull. But it's really interesting that there was all of this upregulation of P53, almost as if it was a way of controlling the chaos. And so if you take all this data and put it on this spectrum of similarity, the elk antler, the velvet, and the cells are actually more similar to osteosarcoma than they are to normal bone. And yet somehow they do this in a photoperiod triggered way and turn off without causing cancer. And I just think there's a ton more work to do there. So I'm going to shut up now. I hope I have told you a story that has been entertaining, but I hope it's also made you think that there is a lot we can learn from studying cancer across species. I hope you'll also think about the ubiquity of cancer and how we really cannot evolve our way out of this, but we can learn what some of Mother Nature's strategies are to control the cheating that is cancer. And this is probably not an audience where I have to make an argument for studying cancer across the tree of life itself. But I do think hopefully this is a reminder that we need to move away from this paradigm of looking at cancer in our chosen species plus a correlative mouse model. And I will shut up now. Joanne has a surgery, and so I'm really impressed you made it for the whole thing. Thank you.
Thank you, Dr. Eward. Fascinating talk. I think we have about 5 minutes. Questions?
First of all, that's was not only incredibly informative, but entertaining, and she said great stories. The elk antler story at the end reminded me of something, and unfortunately, I don't remember all the details, but you might. Apparently, whales are pretty resistant to cancer.
Yes.
And we were recruiting a gentleman a number of years ago from the University of Southern Maine, if I remember, who ended up not coming. That's another story. Who studied cancer or lack thereof in whales. And I remember his talk, it was a decade ago, and he was culturing cells. He would go out and harpoon dead biopsies from whales and culturing them. And he was making an argument that's starting to sound to me similar to what I'm just hearing from you about the elk antlers, about the environment and during growth and some things that are more like cancer, but they don't actually get cancer. Do you know this story? And I'm just trying to put it together.
Yeah, well, I think it's probably more like a storybook with a lot of little stories. We're actually working in this space too. It took us a long time to get some cells and establish some whale cell lines. We're having a very hard time inducing them to form cancer, but I don't think anyone knows why that is. But I'm sure for whales to be whales, they've evolved some kind of strategy to mitigate that. I can tell you one of the parts of this story I find the most interesting is studies of orcas in the Pacific Northwest, which I'm sorry, I tortured some of us at dinner last night talking about this. But if you look at orcas in the Pacific Northwest, female whales live about 40 years longer than male whales. And the reason is they accumulate all these toxins in their blubber, but the females periodically lactate and they consume their fat stores and they actually shed these toxins into their breast milk. And this whole story is so depressing. But it's interesting that males end up dying from the toxins they carry in their blubber. But these are highly carcinogenic toxins, and it's rare, maybe it never happens, that orcas are discovered with tumors. Yeah.
So with the antlers and with the non-rapid growing bone, and it's my memory that with antlers, they actually recapitulate a lot of the initial morphogenetic signaling and patterning that's necessary for even limb induction, et cetera. So do any of those pathways, BMPs, retinoids, sonic, do they actually influence the rapid growth, and are they controlled in a way that does not make them more likely to transform the cells that they're acting on?
So I don't know the answer to that. And based on the reading I did to try to help Nicole through this project, I believe the answer to that is unknown.
Because it seems like another aspect of the signaling that even in a sarcoma, because it's recapitulating at least a little bit of the mesodermal rapid expansion, that could be another handle on.
Right.
And also the differences between the species.
Right. And another question in that space that I have is, you think about bone normally responds in accordance with Wolff's law, that it grows in proportion to the forces acting on it. Mm-hmm. My femur and your femur look alike because we're both ambulatory, not because we're fourth cousins. And yet in deer, the shape and size of their antlers is heritable. So there is something heritable that's driving the size and shape of these antlers that's turned on and off.
Any other questions? Let me check online, see if
Thank you very much.